Hello, everyone. Thank you for joining us today for our last CMDI seminar of the summer. Today, we have Jonathan Humphreys. He's currently a postdoctoral researcher at the National Renewable Energy Laboratory. Jonathan got his PhD and his first postdoc at the University of Nottingham. And today, he's going to tell us a story about a Clostridial strain degeneration, a microbe that is currently being studied for its use in the production of biofuels. Jonathan, thank you so much for coming here, and look forward to hearing you.
Thank you. Thank you very much for inviting me over, or at least inviting me to give this talk. Right, can we see this?
Yes, perfect.
Great. Yeah, so I'll start. My name's John. I'm currently working in Denver in Colorado at the National Renewable Energy Laboratory, but I'm going to be talking today about the work I did in my PhD and my postdoc. So they were over the last sort of 5 years, and it's all about sort of Clostridial strain degeneration. And we're looking at sort of new approaches to a very old problem. So a little bit of background about, about the genus Clostridium. So these are gram-positive, rod-shaped, strictly anaerobic spore-forming organisms. And this is generally what they look like. Some Clostridium, they keep the spores they make within inside the cell. Others, they release it like we see over here. But this is generally a very typical kind of Clostridial cell. There's lots of different species of Clostridia. So we have the more well-known kind of pathogenic species that people may know about, sort of like Clostridium difficile, which causes diarrhea, hospital-acquired diarrhea, which is a big problem in hospitals. Another one is Clostridium botulinum, which makes this kind of deadly paralysis toxin, which most people know as Botox. However, we have many other species. There's the industrial species, which is what I'm more interested in. Some of them are called the acetogens. So these are ones that grow on syngas. So they grow on things like carbon dioxide, carbon monoxide. And this is what I'm currently working on at my job in Colorado. However, my PhD was more focused on the solventogenic ones. So these are sugar fermenters. So they grow on like glucose and fructose, and they make sort of interesting products from this. And these are a couple of examples of those solventogenic Clostridia. So as this is the main focus, a little bit of background about them themselves. They kind of grow by this very unique biphasic metabolism, which what makes them kind of really interesting and what people are interested in using them for. So in the first phase of growth for these organisms, they grow when they're growing exponentially. They kind of make the acids acetate and butyrate, and this is kind of like how they generate most of their ATP. It's basically growing on sugar and making acids. However, as they continue to make acids, it eventually kind of causes the pH of the medium that they're in to drop, and this becomes toxic to them. So to kind of evade this toxicity, they then convert these acids, so they kind of take them back in, and they convert them to the solvents acetone, butanol, ethanol. And this whole process is called ABE fermentation. And this is kind of why we are really interested in them from an industrial point of view, because we can make acetone, all these kind of interesting solvents from a biological means. And these do them completely naturally. And this is sort of an example of kind of like a product profile that you would typically see with these Clostridia, is you get the kind of acids being made over here in the first kind of like 10 to 20 hours. And then eventually when the pH is dropped to a certain point where it becomes toxic, they then start making the solvents. And another really key part of the solventogenic Clostridia is once they start making solvents, they then start making spores. So this whole process is very linked together. Once you are committed to making solvents, you're also committed to making spores, which is going to be important later on. So again, a little bit of background about them, why we're interested in them again. They used to be used kind of throughout the early to mid 1900s for solvent production. So this guy over here, Chaim Weizmann, was kind of the pioneer of it all, the grandfather of solvent production using Clostridia. And he was tasked by the British government to make it during World War I and World War II. And he was basically found that, yeah, we can get bacteria to make all the solvents. This is great. Eventually, they decided that butanol was a more important kind of product to be made in the 1930s and 50s because they needed it for the kind of lacquers of cars. And then around the 1950s, global kind of production of butanol, AB fermentation accounted for about 66% of butanol production and then 10% of acetone production. So this was a huge industry up until about the 1950s. There was a decline, as many of you probably not heard of it or even know about the decline. This was basically because petroleum and using oil, gas, and crude oil and all that kind of thing became far cheaper to make solvents. It was much easier, much cheaper, so then the whole process kind of died away. The substrates as well to grow these organisms became very expensive, and then in 1982, the last commercial plant shut down. There really was this kind of like heyday from the 1900s to the 1950s, and then this eventual decline. We, however, have now got a renewed shift there. We want to start using alternative fuel sources, making alternative fuel sources. There's a big push for things like biofuels, all that sort of thing, because as everybody knows, fossil fuels are running out. And specifically, butanol is seen as a much better fuel than bioethanol. A lot of the production for biofuels these days is bioethanol made by yeast. However, butanol you can put straight in your vehicles. You don't have to make any modifications. It's got much better properties that people would want to use. So that's why we're really trying to look at alternative ways to make it. And Clostridial might be a good way to do that. And there has been some kind of commercial plants, these here, that were built in China, but they're not currently operational. This is mainly due to the fact that crude oil is still kind of cheap, and the whole process isn't economically viable just yet. And another reason it's not economically viable is this whole problem of strain degeneration, this very interesting phenomenon that these bacteria do. And this is basically when the solvent-genic species, they lose the ability to produce solvents and form spores. So these very two key processes that these organisms are associated with doing, they eventually just stop doing for some unknown reason at this point. It seems to occur when the bacteria are repeatedly subcultured, so in these batch fermentations. So what would usually happen is they would basically grow the bacteria in these big fermenters, they would take a small portion and put it in another fermenter, but each time they did that, they found that their product titers were going lower and lower, which obviously isn't good. And then they also found that in continuous fermentations, the problem was also happening. So in both these batch and continuous, we're getting this problem. So it isn't just the fact that you're transferring it, there's obviously other things going on. It can cause cultures to acid crash and therefore die. This is because if they're not, take those acids they originally produced, if they're not converting them to solvent, the pH is just going to go all the way down, and it's going to kill everything. So again, this is a big problem if they're not making these solvents. Much of the work that was done on degeneration was way back in the sort of 80s and 90s, and most people commented on the fact that they saw colony changes. And here's a couple of examples of what they found sort of 1995, so a very long time ago. And you can see they've got these kind of weird colony growths coming across. But this was really to the extent of what people found. Another thing they noticed in the whole kind of degeneration process was in a very well-known strain called Clostridium acetobutylicum. This strain harbors what we call a P-Sol1 megaplasmid. And this megaplasmid has all the solvent genes on it. So if the organism loses this megaplasmid, it's obviously not going to make solvents anymore. So that kind of makes sense. However, in pretty much all of the solvent-genic Clostridia, they have the genes to make solvents on the chromosome rather than on a megaplasmid. So why they degenerate at this stage is very much unknown. There were a couple of ideas back in the sort of 80s and 90s, was maybe it was something to do with the growth rate. So perhaps if these organisms are growing too fast, they won't be able to induce the solvent genes and therefore they degenerate. These guys sort of thought maybe there's a defect in a global regulator or something like that. And then a more recent study kind of linked it to calcium carbonate. However, I'm a little bit dubious of this study. It was a bit kind of sketchy how they did things. But at this stage, these are really the only kind of proposed ideas to why strains degenerate, which doesn't really leave much to think about. So this is kind of where my PhD came in, where I came in, why I wanted to look at it and all that sort of thing. And the sort of aims of my projects, the PhD and then the year postdoc afterwards, was basically to understand degeneration more fundamentally. You know, when does degeneration happen in a culture? How does it happen? You know, these are very unknown things. And at this stage, there had been very limited use of modern techniques. You know, we have all these amazing technologies like DNA sequencing, RNA sequencing, ultra-deep sequencing that nobody ever really used to study degeneration, and it seemed like a really good place to use them. And then once we use these techniques, can we find genes involved in degeneration? You know, there was this idea that maybe a global regulator is involved, but nobody knows what that could be at this stage. And then ultimately, we'd want to prevent degeneration. If we're going to make the use of solvent-genic Clostridia viable, we want to prevent the whole thing going on. But we obviously need to understand it a lot more better than what's known now, just so we can kind of attempt at preventing it from happening. So all the work that I did was done on a strain called Clostridium barynckii, NCI B8052, and we chose this strain for a couple of reasons. One main reason was really the fact that this strain degenerates a little bit more quickly than other strains. We thought if it degenerates a little bit quicker, maybe there's something in that. It's a good place to start when we want to look at the whole process. And another reason we chose it is it did serve as a parent strain to like a hyperbutanol producer, so it does have like industrial relevance as well in case we find anything interesting. And then as we've chosen organism, more of like a side thing, we also thought, well, can we just gain a better understanding of this organism itself? You know, can we find more genes involved in Clostridium barynckii and just get a greater understanding for the wider community? So the whole thing about my project was this old problem of degeneration, this thing that plagued the industry for a long time, but can we address it looking at new techniques? And that was kind of like the whole tagline for the PhD. So basically, right at the start of the project, we wanted to isolate them. You know, if we're going to study degenerates, if we're going to learn about them, we need to find them, we need to isolate them. So I set up this kind of schematic where what we did is we started with a spore stock. So this is basically where you leave the wild type to grow for about 5 to 7 days. It forms the spores, and then what you can do is heat shock it at 80 degrees for 10 minutes. And effectively, this causes any spores to germinate. And what it will also do is it will get rid of any vegetative cells, but more importantly, it will get rid of any degenerate cells, so any cells that haven't been able to form viable spores. This was a good way to start from a purely wild type stock. And we basically plated it out. We then took a single colony. So this was to basically know what the parental wild type genetic makeup was. So we took this single colony, and then we repeatedly subcultured this colony, in a 1 in 10 dilution every 24 hours, and we basically just plated out to see, what was going on. As I showed before, colony morphology was quite important, the things they found, so we thought, okay, maybe we start with this. And this is effectively what we found. So in the 0 subculture, we have these kind of like round and dark, kind of creamy brown colonies, and this is the wild type. This is what we expect. After 2 subcultures, however, we start to see the emergence of these kind of weird colonies here. As you see, they've got these kind of weird outgrowths, similar to the ones that I showed earlier. After 4 subcultures, there's a lot more of these kind of strange colonies coming up, maybe some new ones. By 6 subcultures, there's a whole array now. Very little kind of round and dark wild type looking colonies left, maybe like 1 over here. And then by 8 subcultures, we've got this whole zoo of different colony morphologies. And as you can see, they all seem very distinct from each other. Got different ones here and here, but they're all very distinct from this original wild type. So clearly by subculturing, something weird is going on in the colony morphologies. So we found that there were basically four morphotypes. The first one being the round and dark, which we assumed to be the wild type. The second one we called the dark center with outgrowths, because as the name suggests, these dark center with these kind of like weird outgrowths coming across from it. The third type we called the caved in center, which is kind of these like donut looking colony types. And then the fourth type was this flat and white type, which is these kind of like snowflake looking colonies. And as I say, you can clearly see they're all very distinct from each other, but they all came from an individual single colony, which is quite interesting. And this is a picture I like to include in my talk, just because you can clearly see all the types together on one plate. The round and dark here, caved in center, dark center here, and then the flat and white. So I just like to include it because like I say, you can clearly see that they all are coexisting with each other in a culture, but when you plate them out, they obviously look different to each other. So there's a lot going on in here, but the main thing to get from this is what we did is we basically isolated a load of degenerates, and we started to phenotypically characterize them. And we characterize them in terms of their solvent production, their acid production, and their spore production. These just very basic things that these organisms do. And generally what we found was that the round and dark type, the dark center, and the caved in center, generally these all produce solvents, so the green bars here, produce solvents to a very similar level to the wild type. So as you can see, most of these types did. There were a few outliers here and here that produced a little bit less, but generally these types seem to be very similar to the wild type. We did, however, find that that the flat and white types produce, well, kind of the opposite. They produce significantly less solvents. So most of these types that we isolated produce very little solvent. It's kind of pointing towards the fact that these guys may be the most degenerated out of all the types. But it was kind of interesting to see that although these ones do look similar, they are still kind of performing like the wild type, whereas this one is completely different. When we looked at the amount of spores they made, though, there was a slightly different kind of story, which was a little bit interesting. So generally, although many of those types could make solvents, those ones I showed, they all, except for one here, produce significantly less spores than the wild type. So this is kind of interesting because usually we've always associated spore production and solvent production together, like they're meant to be very intimately linked. So the fact that they can still make normal solvents but make very little spores is quite interesting. And then generally, again, the flat and white type produce pretty much no spores at all. So more pointing towards the fact that this type here is probably the most degenerated out of the four that we've isolated. And again, another interesting thing is like the wild type looking colonies are not performing like the wild type too. So that was kind of interesting to see. So now we'd kind of looked at these colonies, we found that they're kind of acting in their own sort of way. We wanted to kind of have a look at what's going on in the culture dynamics, you know, what's happening in the whole culture with these whole things floating around with each other when we did kind of like a subculturing experiment. So this is effectively what we did is I did that 1 in 10 subculturing every 24 hours. And we found that at each progressive subculture, we're getting a decrease in the amount of solvents that are being produced. So subculturing is clearly affecting the productivity of the culture because it's decreasing. You know, we're going from 100 millimolar of solvents all the way down to sort of 20. It's a big decrease. And then alongside that, we're getting this increase in acid. So this typical kind of degeneration profile. Again, similar to the solvent, spores went down all the way as well. So clearly, subculturing is causing degeneration to occur. What was really interesting, though, is when we started looking at the colony morphologies, their dynamics, and we found something very interesting. It wasn't kind of, there was a lot of things going on in this respect. So in that same experiment, we basically found that the round and dark types, so the wild type looking ones, kind of rapidly decrease towards, and at about 10 subcultures, they hardly exist in the culture. So they are completely going. And they were initially kind of outcompeted by this dark zone outgrowth type. So kind of subculture 0s to 5, you can see that these guys take up around about 65-70% of the population. So this is like the first type to kind of take over for whatever reason. There was, however, you know, the flat and white type that was slowly creeping up. And by the end of the experiment, this type occupied sort of 80 to 90%. So we're seeing these kind of dynamic changes through this whole subculturing process where we have kind of the initial takeover by the dark center and then the final out competition by the flat and white type, which, as we already saw, was kind of the most degenerated out of all the colony types. There was a little increase in the caved in center types, but again, this type didn't really kind of play a role. It seems to be the dark center and the flat and white type. But I just think it's really interesting, really cool to see these like dynamic things going on. And it's not always kind of one type does this, one type does that. So now we've kind of seen that there's colony stuff going on. There's dynamic changes in the culture. Now we wanted to start looking at what's going on the genetic level. So we basically isolated 71 degenerates. So of all of those 4 types, there was roughly about 18 of each. And we whole genome sequenced them all. And we found that there was about 3.3 mutations per degenerate on average that were kind of mapped all over the chromosome. So these red lines are basically where we found a mutation, and they're all over. However, there were kind of these 4 regions that we found had more mutations than the rest of the genome. Now we're starting to see, okay, maybe there are these hotspot regions that are coming up. 2 of these regions were histidine kinases, so C by 0017 and C by 3078. And them being histidine kinase is going to be very interesting, that I'll talk about in a minute. Another region was a hypothetical protein and a transcription regulator called ABRB. And then the final region, which is interesting, was one called supposeaway. And supposeaway is known as the master regulator of solvent and spore production. So if we're getting mutations in this, we're obviously going to affect those 2 processes that we're interested in. So although it seemed kind of obvious that if we get mutations in this, we're probably going to affect it, it was kind of good to see that this master regulator is being affected. And that kind of links to the ideas that people were proposing before as well. And the most interesting thing as well was that many of those flat and white types that I found earlier, the ones that were most degenerated, they had mutations in this BozoAway. So that kind of already fits into the picture of being the most degenerated because they'd lost effectively the ability to make solvents and spores because of supposeaway. I'll give a little of a background on supposeaway because it is a very important protein in Clostridia. Like I say, it's the master regulator of solvent and spore formation, these two late-stage phenotypes that we are very interested in. It has two domains, the protein. It has an N-terminal phosphoacceptor domain here, and then it has a C-terminal DNA binding domain here. And effectively how the protein works is it gets phosphorylated, which causes a conformational change in the protein, which allows it to bind to DNA at places we call OA boxes with this sequence here. And once it's bound to these OA boxes, it can either cause an increase or decrease in gene expression by either recruiting or stopping polymerases. And this is really interesting because what it will do is it will either basically promote the solvent genes or it will kind of repress the acid genes. And this is how the organism switch to these two different metabolisms. This very specific DNA binding domain is highly conserved amongst Clostridia as well, which has this sequence here. So it's very important in Clostridia in general. Even the pathogenic species, they use postdoc as well to do things with their toxin production, all that sort of thing. So it is very important in Clostridia. Now, the activation of the protein is going to be interesting because of what we found. So in the bacillus model, so bacillus are basically aerobic Clostridia. They have a phosphorelay model of how they activate their supposeaway. And effectively what it is histidine kinases will phosphorylate a receiver protein called supposeaway, which then phosphorylates a phosphotransfer protein called supposeaway, which then phosphorylates supposeaway activating the protein. In Clostridium though, or at least in Clostridium acetobactillicum, they found that it was basically a direct phosphorylation model. So histidine kinases would directly phosphorylate supposeaway, causing activation of the protein. Now, this is interesting to us because we found these two hotspot regions which were histidine kinases. So perhaps these histidine kinases are now linked to the activation of supposeaway. So the picture now of this whole degeneration process is kind of pointing towards supposeaway, whether that's kind of direct mutations or perhaps kind of proteins that activate supposeaway or have some involvement in supposeaway. So to kind of test this to see if these histidine kinases were important in supposeaway activation, we did something called clostron insertion or inactivation. This is effectively where you basically chuck a load of DNA into a gene, rendering it functionally inactive. So it's not a complete deletion, it's just a quick way to kind of like inactivate a protein. And we did this for the 0017 and the 3078, the two histidine kinases, and supposeaway. And then we also complemented the genes back on the chromosome to kind of prove their role. And effectively what we found for the histidine kinases were not really much effect on solvent production. So here's the 0017 and the 3078. They were not statistically different to the wild type in terms of their solvent production. The supposeaway mutant basically made no solvents at all. That's to be expected. When we looked at the amount of spores they made, however, this was really interesting because now we see that they made significantly less spores. You know, here's the 0017 and 3078, much less spores to the wild type. And really goodly was that the fact that complementation could restore spore production. But this was really interesting because we had all these natural isolates that I found earlier that produce these kind of phenotypes. You know, they were able to make solvents but couldn't make spores. So clearly these genes are involved in some capacity. But this was interesting to see that we could kind of replicate these natural isolates. And then supposeaway again made no spores, but this is to be expected, and complementation worked as well. But really the take-home message is these histidine kinases are involved in spore production but not solvent production. When we looked at the colony morphology of these as well, we found that basically the 0017 colonies look like the wild type, and many of those round and dark isolates that I got before, they had mutations in 0017. So this is not affecting the morphology at all and making them still appear like the wild type, even though they're not. The 3078, though, you can clearly see looks like the dark center outgrowth type. You know, it really has that chestnut center with the wispy outgrowths. And again, many of those dark center outgrowths type that I isolated were mutants of 3078. And then finally, the Clostridial mutant of supposeaway, again, looks like the flat Y. So clearly, these two genes here are affecting colony morphology in some capacity. And it was really nice to see that we could complement the gene. Once we complemented them back, they went back to wild-type looking morphologies as well. So this is kind of it was really good to see, a really interesting thing to see. So something that we've done more recently, so this is something I pretty much did just before I left my postdoc, and it's very underdone at the moment. I'm just including it because it's like a cool technology. It's something we did called ultra deep sequencing to follow on from kind of the genome sequencing. And effectively what this is you can sequence whole cultures of a population. So take a bit of a culture, you can sequence the whole thing, and you can find when mutations occur within anywhere between kind of 2 to 10% of the culture. So we thought this would be a really interesting way where we could start monitoring when mutations arise within a culture. You know, we've done these subculturing experiments, we've seen the colonies, but when do these mutations happen, and you know what happens when they do? I'm only going to focus on postdoc just because it's a bit more important. And again, this whole thing is still underdeveloped and is in the process of being made into much nicer figures. But effectively what we found was we did a simple subculturing experiment. And after one subculture, we found that solvents decreased here. And when we looked at this ultra deep sequencing, we found that there was a mutation here in postdoc that occurred at this point. So the moment this mutation happened, we got a decrease in the amount of solvents, and this mutation rapidly enriched in the culture. As the darker the red, the more it is in the culture, basically, and that's what we found. So this was really interesting to see. Another example where the same thing happened, we lost solvents pretty much straight away in this example, and again, another mutation, it was somewhere else in supposeaway, but it happened and it enriched in the culture. So clearly, once we're getting a mutation in supposeaway, which may just be a random mutation, it's able to kind of rapidly enrich in the culture. Here's another example as well, where we kind of maintained solvents for a little bit longer, but then we got sort of a decrease here, and this again coincided with, although lots of mutations, it was mutations in supposeaway. We had a few different ones here, but again, once they happened, they enriched in the culture and solvents went down. So clearly, supposeaway is playing a very important role in this whole kind of degeneration process, and this was really, really cool to see that we could pinpoint a mutation that is causing everything to kind of collapse as what it would usually be. So with this in mind, you know, now we're starting to think, okay, supposeaway must have a role in fitness. You know, it must do something to the organism to allow them to grow better or whatever. And eventually, you know, these types are taking over. So from this paper that probably many of you already know, there's a quote which says, cheetahs should do better when they're rare because there are more public goods / cooperators for them to exploit. So now we're kind of getting our kind of cheating hat on and thinking about the sort of roles in this regard. So what we did was effectively a very simple frequency-dependent fitness assay where we mixed a supposeaway mutant with the wild type and just plated them out and had a look what was going on. So we took what I call a natural supposeaway mutant. So this was a mutant that had a mutation, the DNA binding site of the protein, and effectively rendered it completely inactive. The mutant couldn't make solvents, couldn't make spores at all. We mixed it in sort of various initial proportions with the wild type and just calculated the relative fitness based on one of Steve's papers. And this is effectively what we found. So when the proportion of the mutant was very low in the population, its relative fitness to the wild type was huge. It was massive. And you could see this on the plate. You would add a very small amount of the mutant, like 1 to 2%, and it would completely take over the culture within the first 12 and 24 hours, which is really amazing to see. And then as we kind of slowly increased the amount of the mutant in the population, these fitness values began to decrease. And eventually the mutant was less fit than the wild type when it occupied, over sort of 80%. So clearly we're seeing some kind of frequency dependent fitness going on depending on how much is in there. And this was kind of really interesting to see. We repeated the same experiment with a Clostridial mutant. So we had our natural one that evolved from just randomly having a mutation. We then picked a Clostridial mutant, so this was an artificial one. And although it's not as clear on this end, but we are seeing a very similar trend where small amounts of this mutant has high fitness values. These were slowly decreasing and eventually it kind of drops off as well. So clearly, supposeaway is having a role in fitness. To kind of follow on from this, we thought, okay, so fully losing the protein's activity gives a fitness advantage. What happens if we partially lose the activity of the protein? So we took a mutant of supposeaway that was able to make solvents but made no spores whatsoever. So this is why we termed it the partial supposeaway mutant. And again, we found these very similar trends. Very similar trends. It didn't have as high fitness values compared to the natural one, but it was still displaying this frequency-dependent fitness. And then finally, we decided to, okay, what happens if we mix the full zero-way supposeaway mutant with the partial one? Is there some kind of like hierarchical fitness? And that is kind of what we found, is although, again, the trends aren't as nice as the other ones, we found that the full supposeaway mutant gave relative fitness values higher than the partial one when they were mixed together. So the full one would outcompete the partial mutant. So it seems like there is this kind of like hierarchical thing going on where fully losing the protein's activity allows you the most fitness, then before that becomes the partial one, which may explain why we had these kind of dynamic changes in the amount of colonies types that were existing when I did that subculturing experiment, because many of those ones that initially arose were kind of like partial mutants compared to the final types, which were the full mutants. But this was just really great to see that we could kind of show measurably that there was some kind of like fitness advantage going on. So then we wanted to kind of explore this a little bit further. Why are they getting a fitness advantage? Are they just going faster than the wild type? This is something we wanted to look at. So we just did some very simple monoculture experiments just to see, is the mutant growing faster? And it kind of looked like it was growing a little bit faster, the red, but it wasn't too conclusive to us. But something from these experiments became very interesting. We found that when we looked at the CFUs of these organisms, that it actually died. It began to die after 24 hours. So it seemed like it was able to really grow in the first 12 and 24 hours. It was able to rapidly take over, but for some reason, it couldn't sustain itself. And this was interesting. It seemed like it kind of was growing by this kind of get quick rich scheme, where it could outcompete everything, but then was like, okay, I don't know what to do anymore, sort of thing. So this is something we started to explore as well, just to kind of gain a greater understanding of everything that was going on. And we thought, okay, well, if these organisms are, continually making acids, these mutants are continually making acids, and they're not making solvents, perhaps that's probably the cause that is making them die. So we grew the wild type, and we grew this FW7, which is the natural supposeaway mutant. We grew that in these things called pH-controlled fermenters. So these were controlled at pH 6, which should be completely non-toxic to the organisms. They should be able to survive fine. And effectively what we found was the wild type was able to live happily until the end of the experiment, 120 hours, consumed all the glucose. That's what was great. The mutant, however, still died. So we still saw death of this mutant. We were able to kind of maintain it to the end of the experiment, which we hadn't seen before. Usually by 72 hours, the mutant is gone. So we could maintain it, although it had rapidly, massively decreased compared to the higher CFUs. But something interesting we found was that glucose consumption by the organism had stopped. So after 24 hours, effectively, it stopped metabolizing glucose anymore. So clearly, supposeaway is playing some kind of role in this as well, which is kind of interesting because people only see it as this kind of late stage thing where it causes the switch to solvent formation, but doesn't really have any involvement in anything else. But obviously, with this kind of the cessation of glucose consumption, it is affecting that as well. So this was kind of interesting as well to see. So to kind of understand that aspect of it, the role of supposeaway in the early stages, which people have thought it wasn't involved in, we decided to do some RNA sequencing comparing to the wild type. So we kind of took samples in the early, the mid, and the late when it was growing exponentially. And this data is, again, similar to the ultra sequencing, quite fresh, so we're still going through it. But generally, what we found was we had lots of upregulated genes which were in a formate dehydrogenase and ferredoxin subunits. And these are very much involved in redox balancing within the cell. So clearly, by not having supposeaway, the redox going on is kind of all messed up. And we found various other metabolism genes as well. So supposeaway is clearly being pointed towards this as well, alongside the solvent formation too. The downregulated genes we found were sporulation and solvent formation. So this was exactly to be expected. Again, like I say, this is still ongoing. There's still no definitive answer when it comes to this stuff, but I just wanted to include it just to show that we're trying to hit it all at all angles, and we really need to kind of dig through this, but it's been kind of hard since I moved over. So sort of finally, the things to talk about is when we started kind of doing some kind of proof of concept experiments to kind of expand the knowledge of degeneration. And one of them was to adjust the transfer time. So we'd kind of shown that the mutants dying after 48 hours, what happens if we change the transfer time past that point? Can we maintain viability or what? So on the other side of that, we did a 12-hour transfer. So we usually do 24. Now we've done a 12. And we found basically the same thing as the 24-hour transfer, is that we're getting this rapid decrease of solvents. That's to be expected. When we did a 72-hour transfer, however, we were able to maintain solvents for the whole experiment. So this was subcultured 10 times, so every 72 hours, 10 times. And we did lose solvents a little bit, down to 80 millimolar, but throughout the whole experiment, we could maintain solvent production. And this was something we'd never seen before. So basically, by changing this transfer time to a point where these organisms had probably died, we could maintain viability throughout. And again, when we looked at the amount of spores that were produced, these were maintained throughout the whole experiment. So this was the first time, by still doing subculturing, by just adjusting one parameter, we could completely change what we would usually see. And this was cool to see. Another kind of proof of concept one we did was we applied a bottleneck. And this bottleneck that we did was heat shocking. So if you remember, I said at the start, I heat shocked these cultures to kind of get rid of any degenerate cells. Well, this is what we did. So this was a usual 24-hour transfer that we did in a 1-in-10 dilution, but I would heat shock it before. So 80 degrees, 10 minutes, would kill everything except for stuff that have made spores, do the subculturing. And again, what we found was we could maintain solvents throughout the whole experiment. So this was great to see. So just applying this simple bottleneck of heat shocking, we can maintain solvents. And I had never seen this before, solvents all the way to 15 subcultures, pretty much not varying at all. So this was really great to see. And then again, looking at the spore counts, they were maintained too. What was kind of interesting as well when we looked at the colony types of this specific experiment is we found that we basically maintained the round and dark pretty much throughout the whole experiment. And again, hadn't seen this before. And there wasn't these kind of dynamic changes that we saw with the other experiments. You know, we're getting a little bit of the dark center types, which could still produce spores, so that kind of makes sense. But generally, we could kind of maintain the round and dark throughout the whole experiment. And this was really great to see. There was a very little flat and white sort of caved in centers, maybe like a couple of colonies that may have just spontaneously arose. But clearly, by applying these bottlenecks or changing the transfer times, we can affect degeneration or the rate of degeneration in different ways. So to conclude, this really was kind of the first study to underline the genetic causes of degeneration. You know, we found these kind of 4 hotspot regions that we believe are involved in some way. We showed with this ultra deep sequencing that there were like dynamic changes, and that once a supposeaway mutant kind of happened in the population, it rapidly enriched and was able to take over. We've shown that it seems that mutations are in regulators or regulatory cascades rather than enzymes or structural genes. So this is, again, interesting for us. And then, we found more genes involved in the regulation of sporulation in this strain than any other studies. People have looked at it, but they've never really done anything with this organism before. So this was good just to expand the knowledge of this organism on a more fundamental level. We've shown the fitness increases. So, the supposeaway mutants displaying some kind of social cheating. We haven't kind of worked out what that is yet, but clearly by losing the activity of supposeaway, you're able to outcompete other sort of mutants as well, or even the wild type. So clearly this gene is instilling a fitness advantage. And again, when we transfer the times to when this mutant had died, we can kind of maintain viability, or even apply a bottleneck which a mutant of this gene would not be able to survive. And then the next step, obviously, is prevention. Something I can't really do now because I've moved to a different place, but ultimately now we have this knowledge, can we kind of strengthen supposeaway in a way? Can we prevent it from getting mutations? I've got a couple ideas for that, but hopefully maybe one day another student will take over and continue this work. So just to thank my supervisors, Klaus Finzer, Steve Diggle, Alan Cockayne, my friend Alex from the lab. Johnny DeBebe did all the analysis of the sequencing with DeepSeek as well. The BBSRC for funding me and the SBRC in Nottingham for hosting me. And thank you all for listening. Do you have any questions?
Thank you so much, John. Very interesting talk.
Yeah.
So we have a couple of questions. I just want to remind everybody for asking questions, you can either write your name and you can ask your question directly. You can write your name in the chat section. And a couple of people already wrote their question that I'm going to read out loud and
Okay.
John can answer. I think Max had a couple of questions. One was about the different colony morphologies that you saw, and he's asking, can it be an example of phase variation? And I think his second question was dependent on the status of the cells, that at different stages they're producing different levels of solvents. And he's asking about the redox status of the cells.
The phase variation, I don't think so, because these you could, see with your own eye, and a lot of these pictures I just took with like a phone camera. So I don't think it was anything to do with that. And you could, once I made stocks of these, you could revive them and they would still look like that as well, depending on whatever kind of media you put them on.
And I think the second one was, did you check the redox status of the cells? It was just, I think, about when you were mentioning the expression levels.
No, we have, that was something we didn't check. We hadn't checked, but it's something that could be done for sure.
And Steve is asking, could you explain how the supposeaway mutant might be cheating? What's it cheating on?
So one of the theories I had was it was kind of like, you got these public goods. Well, it was more like a public burden. So what it was doing was kind of continually producing acids, because that's what all it does. It doesn't make solvents anymore. And then the cells around it were then taking these up because the pH was really dropping. This is one of the theories. So instead of them they were taking up the acids, which the mutant was making and was growing really fast. And that was kind of slowing them down because they were having to reassimilate the acids rather than the mutant doing that itself. It was kind of like a burden to them. That was kind of one of my theories.
But, okay. So there are...
Because making acids is the way these organisms produce the most amount of ATP. So they make the most amount of ATP by converting to acids. So therefore, if they just make acids and let the other organisms or the other cells in the culture to take them up, they won't be making the amount of ATP, whereas these ones will. This was sort of one of my early theories kind of thing. And that's why it's more of a public burden than a good. They're not exploiting. Yeah. They're kind of making sure they do other stuff to do them, kind of thing.
Interesting. I think, James has a question. And Steve is saying public burden, new phrase. So I think you can see it. James, can you unmute yourself? Or?
Yeah, sure. You might want to mute yourself because otherwise it'll echo. I'm stood right next to Shada, but Shada's just there. The public burden, does that make any different predictions from a public good? A public good, you'd expect different population structures to have different effects on the fitness benefit to cost. So what different predictions would a public burden have over a public good?
I suppose a public good is usually something you have to make genetically. You have to make a protein, you have to go from that way, and then you're putting it out there. Whereas this is something that they're not taking something up, they're kind of putting it out there, and that's what they're doing to exploit other things. Instead of people taking things in to exploit, it seems like these ones, my theory is they're putting stuff out to exploit, to kind of slow other ones down by kind of, making them take the acids kind of thing. That's sort of what I'm trying to get. It's quite, I don't know, it's something I've been thinking about for a while, but haven't quite come up with the best words to say how it works.
So in a way, it's similar to like an antibiotic. They made something that's impacting the ability of another bacteria to survive in the environment. It reduces fitness.
Yeah, because this acid production will lower the pH, and that lowering the pH triggers organisms to reassimilate. So usually the pH lowering triggers supposeaway to start the solvent production. But if they can't do that because they don't have it, other organisms will do that to kind of maintain the fact that the whole culture should try and survive. Sort of thing.
See, I think it's not. Steve, as Steve's about to say, it's coercion. It's not. It's not definitely not coercion.
Is it coercing the wild type into
It's
doing
definitely
something?
not coercion because it hasn't evolved because its ability to coercion has a definitive meaning. It hasn't evolved to do this benefits. It hasn't evolved to do this action, so it's definitely not coercion.
That was in the paper title, surely it was.
We should change the title because it's not coercion. Coercion's been very well defined. I was about to finish to say I think I haven't got this straight in my head exactly what it would be, but I think there would be different predictions if your idea is right, if that's what's happening, then there would be different outcomes and there's different predictions you could make that you could actually test and say, okay, is this the interaction that's occurring in the system? Much like we could test public goods production, like Diggle did with Sophie. There's a fitness benefit to group size. You can add the molecules back in at certain groups. It becomes a benefit.
I think that was something we were looking at doing, where we would effectively spike the culture with acids, because we can just add butyrate or acetic acid and spike it and see do we get this whole shutdown by doing that. But again, something that wanted to be done but didn't get done in the time. So
yeah, I'll yield my time back to Diggle.
No, I'm done. Nice talk, John.
Thank you.
I'm sorry, I'm muted. Sorry. I have a question about, you get lots of postdoc, and the earlier they arise in the population, the frequency goes higher.
Yeah.
From your deep sequencing analysis.
Yeah, basically, once we found a mutation, that's when everything went, kind of thing. Yeah.
So, from my memory of how postdoc gene relate, so I go back. So, is there any effect in germination of the spores that I know that the postdoc don't sporulate, but in your other mutants, if they didn't have any postdoc mutation, was there any changes in germination level?
What, as in if they had mutations in other genes?
Mm-hmm.
Yeah, so if they had mutations in those histidine kinases that I found, they would have sort of 50% reduction in spore production.
What about germination from those?
Yeah, so the same thing. The test is you grow them up, you heat shock them, and then you would see, compared to the wild types, the wild type makes 10 to the 8. These guys would make 10 to the 5 kind of thing when you germinate them.
Okay. Okay. Yeah. But the spores are still normal.
Yeah.
Okay. Interesting.
So that was kind of, they basically would just make less spores, but they would still act the same way. So I would test them after germinating them, and they would still be the same. They would just make less spores. Kind of thing.
Okay.
Yeah. So it wasn't, because that was one of the things I originally thought was it just a mixed population or something? But no, these were single colonies isolated, tested, and then, so no, they still perform the same way.
Okay, that's great. Interesting, very interesting. Lots of things, lots of questions to be answered.
I have a quick question about the megaplasmid. So you have this piece of DNA that seemingly includes all of these wonderful genes that make make an industry, right? Because that's basically the pathway from acid to solvent.
But that's only on one. That's only one organism has a megaplasmid. Generally, they all have it on the chromosome. It's only one of them. Yeah.
How this may be a silly question, but how people just try to put that somewhere else in another organism that is just well characterized, and you know the genetics, and you know how you basically can you do it in coli?
So they have done that. They've taken the solvent genes from acetobasilicum, which is on this megaplasmid, and they have put it in E. coli, and they made butanol production. But the problem with putting on E. coli and stuff is toxicity. So these organisms have obviously grown to be more resistant to solvents, because solvents kind of destabilize the membrane of these organisms. With E. coli, and these are gram-positive as well, so they're a bit more stronger than E. coli. So they got E. coli to make it, but I don't think it was very happy. So. And that's kind of like the whole thing about it, is that these organisms are adapted. They've learned how to look after solvents much better.
That's awesome. Because I was thinking maybe you put it in an acid tolerant gram-positive, but then these things are solvent tolerant, which is an interesting
because these solvents would basically cause membrane fluidity, and then everything kind of just goes.
Can I ask another question? So if you were going to go to kind of a industry with this, with your findings, what would you recommend that they should do to sort of prevent this acid crash problem? I mean, does it open any doors with that you think might make a difference?
So one of the big things I was looking at doing before I left was kind of linking supposeaway to potentially an essential gene. So if you link supposeaway with an essential gene, that can't be lost, therefore you can't lose supposeaway. So therefore you should be making solvents all the time, And that was either
times to passage and things like that?
Pardon?
What about different times to passage and things like that?
Yeah, the times would work. Yeah, the transfer times, that was something, but that comes into the cost. If you're now changing passage times, the cost is going to increase because you have to wait longer, all this sort of stuff. That's why. And like heat shocking every time is obviously works, but now you're adding an extra step which costs more money, So generally industry wants these continuous processes, but if we're having to change things, it's not going to be good. So that's why I thought if I linked this whole supposeaway and strengthened the gene in some way to prevent it from mutating, maybe that would be a way to kind of prevent degeneration from happening.
I just make sure that there are no questions. Well, thank you so much for joining us. It was good to see you after all these years and see how your work has developed. And once more, I want to thank everybody from our organizing team and everyone in CMDI that gave us the opportunity, and all of the people who joined us during this summer. Juan, Jennifer Rattray, and Jennifer Farrell helped to organize this. Thank you again.
I'd like to add to that and just to congratulate you all for what's been a really good and successful summer series. I think we've all had, I think everybody you've had talking has given really good, interesting talks. I think it's shown this format can work in very difficult times. All the speakers have done a great job. We've had good attendances. It's great you recorded them. So I think, I don't know whether anybody's thanked you guys yet, but I'm thanking you now. So I think you've done a great job.
That's all good. Especially, again, I want to thank the whole team, Juan, Jennifer Rattray, for making all of the slides, all of the flyers, and Maria and everybody else. And just enjoy the rest of the summer. And people at Georgia Tech, we will get back to you for new semesters. Thank you. Bye.
Thank you very much.
Thanks, guys. Bye.
Juan, if you have anything to say, would you want to add anything?
No, thanks everyone for showing up. It was awesome. We'll keep doing this in the fall. So yeah, stay tuned. Bye.
Bye-bye. Thank you.